Mangrove forests are nature's coastal guardians, found where land meets sea across the tropics and subtropics. Their tangled roots hold shorelines in place, protect coasts from storms, and store massive amounts of carbon. Beneath their twisted roots, there are bustling cities for life, home to crabs, snails, and fish. Every mangrove has its own community. Crabs shredding fallen leaves, snails grazing on algae, and fish hiding among roots. Despite their diversity, each species has a unique role, and if one disappears, the whole ecosystem feels the change. Professor Thibodeau and his team at the Chinese University of Hong Kong set out a tricky question: who eats what in the mangrove ecosystem? For years, scientists thought that many mangrove animals had overlapping diets, eating the same things. But we suspected they might actually feed in very different ways. Thus, we studied their trophic position. Basically, each species' location in the food chain. We looked especially at most abundant species like sesamid crabs and gastropods. By studying their diets, we could reveal how energy and nutrients like nitrogen flow through this complex web of life. And that's vital for conservation. If key species have unique roles, losing one could collapse the system. Mapping who eats whom in mangroves is not easy. Traditional tools like looking at stomach contents or using conventional isotope technique can't tell apart nitrogen sources when they're mixed. Crabs and snails might eat mangrove leaves, algae, or even microorganisms, all of which leave similar chemical traces. So, our approach is to use a new detective tool, compound-specific isotope analysis of amino acids. Instead of just looking at the whole organism, we zoomed in on specific amino acids, the tiny building blocks of protein. Some amino acids keep the nitrogen signature from the original food source. Others change as they move up the food chain. By comparing them, we could finally separate what came from mangrove leaves, while what came from marine algae or microbes. Crabs, especially the leaf-eating sesamid crabs, weren't relying only on mangrove leaves as most researchers believed. Their diet was a blend of mangrove leaves and microalgae, with anywhere from 8 to 50% of their nitrogen coming from mangrove material and the rest from marine sources. Snails, or gastropods, told a completely different story. They drew almost all their nutrients from marine algae and microorganisms, not from the mangrove trees. Even crabs that looked almost identical had distinct diets. Two fiddler crab species feeding side by side used entirely different strategies, one scraping algae from the mud, the other eating tiny marine animals. And the so-called mud-eating crabs actually occupied very different spots in the food web. One a low-level grazer, the other a higher-level consumer. These findings showed that mangrove species aren't redundant copies of each other. Each has its own niche, its own way of recycling nutrients through the ecosystem. As can be seen, mangrove animals that look and act similar may actually play very different ecological roles. It means that mangrove ecosystems are less replaceable than we thought. If one key species disappears, there may be no other creature that can fill its role, making these forests more fragile than they appear. So, protecting mangroves is about preserving an entire invisible network of life beneath the muddy surface.